Gene therapy of sickle cell disease. Sickle cell anemia is caused when erythrocytes, or red blood cells, become sickle-shaped. This sickling prevents the hemoglobin inside the cell from carrying oxygen, which can lead to anemia. The sickling can also cause the cell to clump and aggregate in low-oxygen environments, causing organ damage, inflammation, and ultimately organ failure. Sickle cell disease is caused by a point mutation in the 6th codon of the hemoglobin gene. This mutation changes a glutamine to a valine, effectively changing the charge of a section of hemoglobin. In low oxygen environments, such as in the kidneys, hemoglobin goes through a conformational change, which exposes a hydrophobic pocket. A mutant hemoglobin will then bind its valine to the newly exposed hydrophobic section. The protein forms clumps of itself through this mechanism that will eventually sickle the red blood cell. The current method used to cure sickle cell disease has been to transplant a matching donor's hematopoietic stem cells into the marrow of a patient. This is only available to 14% of patients whom have family members that are matches, leaving the rest of patients to rely on medications and blood transfusions. Another method that has been recently devised is to harvest the patient's own hematopoietic stem cells and then insert the correct beta-globin gene with a viral vector. These transformed cells can then be transplanted back into the patient, in which they can differentiate into erythrocytes with the functioning hemoglobin gene. Here are some examples of cassettes that have been proposed to be inserted into stem cells over the last decade or so. The most recent proposal, which is in clinical trials, being at the top, and the least recent being at the bottom. Something I would like to point out are the red sections in the top 3 cassettes, and the light blue on the bottom. These represent hypersensitive sites that were found 40 to 60 kilobases upstream of the hemoglobin gene that promote the differentiation of stem cells into red blood cells. By adding these factors, genetic engineers were able to promote erythrocyte formation. You may also notice many different forms of the hemoglobin gene, with the naturally found hemoglobin gene being at the bottom. The reason natural hemoglobin is a poor anti-sickling compound is because it still forms the hydrophobic pocket that valine can bind to. Meanwhile, in utero, gamma globin contains a gamma chain that blocks this hydrophobic pocket. This hemoglobin variant is no longer produced in adulthood and has too high of an oxygen affinity to properly function outside of the womb. To compromise, a genetic variation of hemoglobin was created with this gamma chain over the hydrophobic pocket, and then edited further to prevent homologous enhancers and repressors from affecting the gene. Originally, retroviruses were used to deliver the gene of interest, but they proved to be a failure due to their inability to hold enough genetic information. They also had a tendency to insert near transcriptional start sites, which would disrupt homologous gene expression. Instead, a vector designed with HIV as a template called lentiviral vectors were used. They would insert more randomly into the genome and could carry a robust amount of genetic information. The production of the vector also involves a 4-plasmid system, which prevents the virus from being able to insert its own replication machinery into the host, preventing viral reproduction. Currently, only a single 13-year-old boy has made it through clinical trials and has been free of all signs of sickle cell disease for over a year. 2 other clinical trials are ongoing with slightly different hemoglobin genes and are scheduled to release their findings in late 2017, of which we are eager to receive.